Welcome back to the deep dive. Today, our mission is to give you a real shortcut into um, what might be the most disruptive breakthroughs happening in pharma right now.
We're looking at early-stage drug discovery.
Exactly. We're tackling the biggest choke point in the whole process. First, the sheer inefficiency of finding a good initial hit, and second, the difficulty of knowing if that hit is even worth pursuing.
It's a huge area of failure, The whole pipeline, from finding that first molecule to optimizing it, it's incredibly slow, it's expensive, and more often than not, it just ends in failure years later.
Right.
But the sources we pulled together today, they detail truly massive computational breakthroughs that are fundamentally changing the landscape. They're making things that were impossible suddenly seem, well, accessible.
So let's define that core problem for everyone. The industry standard has always been high-throughput screening, right, HTS. You basically test millions of different molecules against a protein target, and you just hope one of them sticks. But the numbers, they're just brutal.
Oh, they're terrible.
We're talking about finding an active molecule, a hit, maybe 1 or 2% of the time. You're just burning through so much time and money on compounds that do absolutely nothing.
And that's for the easy targets. That low success rate, it just gets exponentially worse when you start tackling what the industry calls intractable targets.
What does that mean exactly?
Well, think about really flexible immune receptors like TREM2 or these super complex slippery surfaces where two proteins meet protein interfaces. For those kinds of targets, traditional HTS is barely better than just guessing.
Okay, so let's say you get lucky. You beat the odds, you find a hit, you're immediately facing the second bottleneck.
Yes.
So many drug programs fail later on because the researchers focused on the wrong thing. They prioritized binding affinity, basically how tightly the drug grabs onto its target, over something called ligand kinetics.
Specifically, residence time, or RT.
Which is what?
It's how long the drug actually stays bound once it gets there. High affinity looks great in a lab test, but if the molecule just pops right off again in the body,
It can't do its job.
it can't sustain its effect. And predicting that residence time, that has been a computational black hole for decades.
So our sources lay out these two really distinct solutions. One uses AI to tackle the front-end problem, figuring out what to test in the first place.
And the other uses a new kind of physics-based simulation to tackle the back end, predicting the quality of that hit, that residence time with just incredible speed.
Let's name them. We're diving into HTS-Oracle, which is the AI system, and Koffee Unbinding Kinetics, which is the simulation approach.
Right. And together, they start to look like a true end-to-end computational pipeline.
Okay, let's unpack HTS-Oracle first. The whole point here is to avoid testing thousands and thousands of useless compounds. How did they build an AI that's smart enough to predict hits for targets that, have stumped scientists for years?
Well, its strength is really in its hybrid architecture. It's a multimodal deep learning platform, which means it doesn't just look at a molecule in one way. It uses two powerful streams of information at the same time.
Okay, walk us through those two streams.
So the first stream is molecular language modeling. It uses a tool called cheminformatics. And the best way to think about this is that it treats chemical structures not as static pictures, but as sentences.
That's a language.
Exactly. It processes these structures using something called smile strings, which are just text-based representations of a molecule. So it learns the grammar of chemistry. It understands how different atoms and bonds relate to each other in a deep way.
So it's literally reading the molecule. What's the second layer on top of that?
The second layer is more traditional, validated cheminformatics. It uses a toolkit called RDKit to pull out features like Morgan fingerprints.
Which are like maps of specific chemical features.
Right, maps of features and connectivity. And on top of that, it adds 15 key physical-chemical descriptors, things you'd expect like molecular weight, solubility, metrics that help define if a compound is drug-like.
That feels very complimentary. You get the deep, almost intuitive understanding from the AI language model, but it's grounded by these classic quantifiable metrics.
And it's all wrapped in an ensemble of 15 submodels to make sure the predictions are robust and stable. But here's the real genius of the system, I think.
Go on.
It's explicitly designed for prospective screening, for predicting future results. And to do that, it incorporates all the negative screening data, all the millions of compounds that failed.
Wait, so conventional methods just throw that data away?
They just discard it. They only look at the 1% that worked.
So they're tossing out 99% of the potential learning opportunity.
Precisely. By training on the negative data, HTS-Oracle doesn't just learn what works. It learns the very specific reasons why the vast majority of molecules don't work. It understands the boundaries of the chemical space so much better.
Okay, so the sources detail the case studies, and they really didn't pick easy ones. They went after 3 of these intractable targets.
Right. TREM2, which is tied to neural inflammation and diseases like Alzheimer's. Then CHI3L1, which is notoriously hard because it's linked to intrinsically disordered proteins.
And CD28, a big protein-protein interaction target for cancer immunotherapy. These are all incredibly difficult.
They're difficult because they don't have nice, neat little pockets for a drug to bind to. They're flexible, they're complex, and existing hits are just too weak to be useful. HTS-Oracle was designed to make them druggable.
All right, let's get to the results. What kind of efficiency gain did this AI actually deliver?
The numbers are pretty staggering. For TREM2 and CHI3L1, HTS-Oracle reduced the number of compounds that needed to be physically tested in a wet lab by more than 99%.
99%.
More than that. And for the really tough CD28 target, it was a 70% reduction.
That's a massive savings in cost, time, everything. So for TREM2 specifically, what did that look like?
Okay, so for TREM2, they started with a library of over 12,000 compounds. The AI told them to test just 25.
25.
25. And the result was an 87.5-fold enrichment compared to what you'd get from a normal screen. The hit rate jumped from a pathetic 0.32% to an incredible 28%.
An 87 times better chance of finding something useful for less than 1% of the effort. But was the quality of the hits any good?
Much better, actually. The best compound they found had a binding affinity of 6.46 micromolar. The previous best-known ligand for that target was way weaker, at 22.4 micromolar, so it's a huge improvement in both quantity and quality.
What about CHI3L1, the disordered protein target?
That was the biggest win. It achieved a 176-fold improvement in screening efficiency, a 37% hit rate compared to the usual 0.21%.
176 times better. I mean, if you ever needed proof that AI could redefine this process, that's it. And CD28, that was the stress test, right? The data set was really sparse.
It was a huge stress test, yeah.
Yeah.
Only 2% of the training compounds were active. But even with that challenge, HTS-Oracle gave them an 8.4-fold enhancement in the hit rate. And they confirmed 2 of those hits functionally inhibited that key protein-protein interaction.
So HTS-Oracle really works. It takes you from a needle in a haystack to, well, finding a high-quality pin in a small pile of other pins, and it's open source.
Which is huge. It really democratizes this initial step of drug discovery.
Okay, but as we said, finding the hit is just the first part of the war. Now we have to shift to that second bottleneck, the kinetics. We have a good hit, but is it the right hit? We need to know its residence time. Why were the old computational methods for predicting RT so bad? What was the fundamental problem?
It's all about a massive timescale mismatch. A drug unbinding from its target. That's a process that happens over seconds, minutes, even hours.
Okay.
But the computer simulations we use, called molecular dynamics, or MD, they simulate tiny fractions of a second, nanoseconds.
So you're using a stopwatch built for a 100-meter dash to try and time a marathon. It's just the wrong tool.
It's fundamentally the wrong tool. And to try and bridge that gap, researchers had to use these incredibly complex, computationally expensive, enhanced sampling techniques.
Which sounds slow.
It meant a single simulation for one molecule could take several days of GPU time. You simply can't screen thousands of compounds that way. It's impossible.
And this is where Koffee Unbinding Kinetics comes in. How did they get the speed needed to make this a high-throughput tool?
They completely reformulated the physics of the problem. Instead of trying to run a simulation through time to watch the whole unbinding event happen, Koffee uses an energy-resolved optimization process.
Energy resolved? What does that actually mean?
So instead of asking how long until it unbinds, it asks a different question. What's the lowest energy barrier the molecule has to climb over to get out?
Okay.
It quickly finds those low-energy escape pathways and calculates the activation energy, that barrier height.
And once you have that energy value, you can use the Arrhenius equation to link it directly to the residence time. So they sidestep the whole time problem by focusing only on the energy landscape.
That is the core breakthrough. And the result is an unbelievable speed gain. Koffee is 3 to 5 orders of magnitude faster than the old MD-based methods.
Orders of magnitude, what's the actual time?
A single simulation runs in about 1 GPU minute on cheap, off-the-shelf hardware.
From days per molecule down to 1 minute. That just completely changes the game. You could test thousands of compounds a month.
It makes kinetic screening a real routine, high-throughput step. and they validated it across a really diverse set of hard targets.
So what were some of those highlights? They tested all sorts of things, right?
Yeah, they showed strong predictive performance on 6 very different targets.
Yeah.
Everything from big macrocycles to metalloproteins to complex GPCRs. For one target, TTK, they got a correlation of 0.80 between their prediction and the experimental data. That's very strong.
And they pushed it even further, didn't they? They looked at tiny changes in the protein itself.
Yes, single-point mutations in the binding site. This is critical for optimizing a drug. And Koffee correctly predicted the kinetic effects of those mutations in the M3 receptor, a task that, in a previous study, took 182 GPU days to do with MD.
That's incredible. So to really prove it wasn't just, getting lucky, they ran a huge scale test.
They did. Across 332 different ligand-protein complexes, it maintained a really robust ranking performance. But here's the most important finding from that test.
Okay.
Its predictions were completely uncorrelated to the ligand's molecular weight.
Why does that matter?
Because it proves the model isn't just cheating. It's not just saying bigger molecules stick longer. It's actually learning the complex, non-trivial physics of the unbinding process itself.
And did it succeed at the ultimate goal? Could it pick out the winners, the long-acting compounds?
It did. It could classify binders with a residence time over 1 hour with a very high accuracy score. So it absolutely proves its ability to prospectively prioritize the best compounds before you ever have to make them in a lab.
So let's connect the dots. What does this all mean for the future of drug discovery? You have HTS-Oracle on one end.
Dramatically increasing your chances of finding a hit for a tough target by up to 176-fold.
And then you have Koffee on the other end, dramatically speeding up the assessment of that hit's kinetic quality from days down to a single minute.
Together, they create this new, streamlined pipeline where targets that were once considered chemically inaccessible are now completely tractable. Researchers can move on from just brute force screening.
It's a huge leap in specificity. You can now prioritize compounds based on a real driver of efficacy, like residence time, not just simple affinity.
Exactly. And both of these platforms are built for real-world, resource-efficient use. The next step is obviously full integration, where HTS-Oracle finds a hit and Koffee immediately filters it for kinetics, creating this optimized discovery engine.
So think about the implications here, both scientific and economic. You have a process where thousands of wet lab experiments get reduced to just 25 AI-guided ones, and where simulation time drops from days to 1 minute per molecule. A provocative thought we want to leave you with today is this. How will this incredible new speed shift the entire focus of medicinal chemistry away from brute force screening and toward complex, precise kinetic optimization right from day one of a project? And what new biology will that finally allow us to drug?